Microfluidic systems are rapidly transforming space exploration by enabling a host of experiments and resource management techniques that were previously unthinkable in a microgravity environment. In space, where gravity no longer governs fluid motion, forces like surface tension, capillary action, and even electrokinetics become the dominant drivers of fluid behavior. How do these devices work without gravity? The answer lies in carefully engineered microchannels and surface coatings that manipulate these alternative forces to control the flow, mixing, and reaction of liquids at the micro scale. One fascinating example is the use of microfluidic devices to study cellular behavior in orbit. On the International Space Station, researchers have deployed chips that allow for the continuous observation of cell cultures, enabling them to analyze how cells proliferate, differentiate, or even undergo programmed death in a microgravity environment. For instance, experiments on blood clotting have utilized microfluidic chips to mimic blood flow through tiny vessels, revealing unexpected changes in coagulation dynamics when gravity is absent. Could these insights lead to better medical treatments for astronauts? Absolutely. By understanding how cells respond to microgravity, scientists can develop countermeasures to protect crew health during long-term space missions. Another compelling application lies in resource recovery. Microfluidic systems are being adapted to recycle water and extract nutrients from waste products aboard spacecraft. These devices use a combination of microscale filtration, chemical reactions, and phase separation techniques to purify water for reuse. One experiment on the ISS focused on processing urine through microfluidic membranes, where capillary forces drive the separation of water from contaminants. How is this beneficial for space missions? The ability to efficiently recycle water reduces the need for frequent resupply missions and is crucial for sustainable long-duration travel, such as missions to Mars. Scientists are also leveraging microfluidics for incipient chemical analysis of extraterrestrial materials. For example, microfluidic platforms have been designed to analyze lunar and Martian regolith, examining soil samples for water content, mineral composition, and even organic compounds that might hint at past life. These chips integrate chemical sensors with microreactors to conduct assays right on the planetary surface, offering rapid feedback without the delays of sending samples back to Earth. How does this advance planetary exploration? It enables real-time decision-making, allowing rovers or landers to adapt their exploration strategies based on immediate analytical results. Moreover, microfluidic devices are revolutionizing diagnostic capabilities in space. Compact labs on a chip are now capable of performing PCR amplification, immunoassays, and even protein crystallization experiments. For instance, microfluidic PCR systems have been used on the ISS to detect viral or bacterial DNA, providing critical health monitoring for astronauts. How do these systems overcome the challenges of space? By miniaturizing and integrating complex laboratory processes into a single portable device, these systems reduce contamination risks and offer rapid, on-demand diagnostic results. Unexpected applications continue to emerge as well. Researchers are investigating the use of microfluidic devices to study the behavior of fluids in conditions that simulate the formation of interstellar dust or the aggregation of particles in protoplanetary disks. By replicating these phenomena on a chip, scientists can gain insights into the early stages of planet formation. Isn't it fascinating to think that tiny channels on a microchip could help unravel the mysteries of how solar systems come into being? The answer is yes. Through precise control over fluid dynamics at the microscale, microfluidic experiments can mimic processes that occur on astronomical scales. In essence, microfluidics in space exploration is a prime example of how interdisciplinary innovation can overcome extreme environmental challenges. By harnessing forces that dominate in microgravity, these devices enable a wide range of applications, from life support and resource recycling to real-time planetary analysis and advanced biological research. Each breakthrough not only pushes the boundaries of what is technologically possible, but also opens new avenues for understanding our universe. The next time you gaze at the stars, consider how the tiny engineered channels in microfluidic chips are making giant leaps in space science possible. Microfluidic devices are uniquely suited for space exploration because they require minimal reagents, offer precise control over small fluid volumes, and function efficiently in microgravity. Traditional fluid dynamics behave unpredictably in space due to the lack of buoyancy-driven convection and sedimentation. Microfluidic chips overcome these challenges by utilizing capillary forces, electrokinetic transport, and surface tension-driven flow to manipulate liquids in controlled environments. The International Space Station used microfluidic devices to study crystallization processes, protein folding, and bacterial behavior in space. This is like using a pipette to transfer liquid in a zero-gravity environment rather than relying on gravity-fed systems like traditional laboratory setups. Astronauts experience immune system dysregulation, altered gene expression, and increased susceptibility to infections during long-duration spaceflight. Microfluidic Labanat chip (LOC) devices allow real-time monitoring of blood biomarkers, immune responses, and bacterial growth in space with minimal sample processing. These systems enable rapid health diagnostics, helping astronauts manage medical conditions in deep space missions without needing Earth-based laboratories. NASA has tested microfluidic chips for detecting inflammatory cytokines in astronaut blood samples to study immune function in space. This is like having a miniature hospital lab on board a spacecraft, performing real-time health checks without bulky equipment. In space, fluid behavior differs significantly from Earth due to the absence of gravity-driven flow mechanisms such as sedimentation and convection. Microfluidic devices leverage surface tension, capillary forces, and electrokinetics to manipulate fluids with precision, allowing researchers to study phase separation, droplet dynamics, and bubble formation in microgravity. These insights improve spacecraft fuel management, life support systems, and materials science experiments. The ESA's Fluid Science Laboratory on the ISS has tested microfluidic systems to understand liquid-solid interactions and phase transitions in microgravity. This is like learning to pour a drink in zero gravity, where liquid clings to surfaces and moves in unexpected ways unless precisely controlled. Sustaining long-term human presence on Mars or the Moon requires efficient food production, including growing plants in space. Microfluidic bioreactors provide controlled environments for studying plant root growth, microbial symbiosis, and nutrient uptake under altered gravity conditions. These miniaturized systems optimize water and nutrient delivery while reducing waste, supporting sustainable extraterrestrial agriculture. NASA's Veggie experiment on the ISS has integrated microfluidic nutrient delivery systems to grow lettuce, radishes, and mustard greens in microgravity. This is like replacing large farm irrigation systems with precise droplet-based nutrient dispensers for maximum efficiency. Future space missions aim to extract and process local resources such as water and minerals from lunar or Martian soil to reduce dependency on Earth resupplies. Microfluidic systems can analyze and refine extraterrestrial materials, separating valuable elements from regolith through electrophoresis, ion exchange, and capillary-driven extraction methods. These techniques improve resource utilization for fuel production, life support, and construction materials. NASA has proposed microfluidic-based ISRU devices to extract oxygen from lunar regolith and analyze Martian soil for water content. This is like using a portable water filter in the wilderness to purify and extract essential resources from the environment. Astronauts on deep space missions cannot rely on regular medical supply shipments, making on-demand drug synthesis a necessity. Microfluidic reactors enable continuous flow synthesis of pharmaceuticals, allowing the production of essential drugs in microgravity with minimal reagents. These systems facilitate personalized medicine by tailoring drug formulations based on real-time astronaut health diagnostics. Researchers are developing microfluidic reactors that can synthesize antibiotics and painkillers on demand aboard long-duration space missions. This is like having a portable pharmacy in space, where medicines are created as needed rather than stocked in bulk. Water is a critical resource in space, and recycling is essential for long-term missions. Microfluidic filtration and purification systems enable real-time water analysis, detecting contaminants at ultra-low concentrations and purifying water efficiently. Advanced membrane filtration, electrochemical sensing, and biofilm detection help ensure a safe water supply for astronauts. The ISS employs microfluidic-based water quality monitoring systems to assess drinking water purity and detect microbial contamination. This is like a smart water filter that not only purifies water, but also instantly alerts users about contaminants. Microfluidic fuel cells offer an efficient way to generate power in space by utilizing controlled electrochemical reactions. Unlike traditional batteries, these fuel cells can operate with minimal waste, are highly scalable, and function efficiently in the vacuum of space. Their miniaturization allows integration into nanosatellites, rovers, and space habitats. Microfluidic hydrogen fuel cells are being explored as alternative power sources for deep space probes and Mars exploration vehicles. This is like replacing large diesel generators with compact renewable energy cells that produce clean electricity. The next generation of space microfluidic systems will incorporate artificial intelligence, AI, for autonomous data analysis and adaptive control. AI-driven microfluidic platforms can dynamically adjust experimental conditions, optimizing biological studies, chemical synthesis, and material fabrication in space. These systems will enable fully autonomous extraterrestrial research without direct human intervention. NASA is developing AI-assisted microfluidic biosensors that monitor astronaut health in real time and adapt diagnostic protocols accordingly. This is like having a self-iterating laboratory that continuously adjusts its experiments based on real-time results.